So the 3 steps that we're going to talk about, and we're actually just going to talk about the first one, in the breakdown of sugar, the 3 main steps are going to be glycolysis, then the Krebs cycle, there's a step in between, and then the electron transport chain at the end. So we're going to start with the first step, glycolysis, and I mentioned this yesterday. Glyco refers to what? Sugar or glucose, and lysis means to break. So literally, glycolysis is just the splitting of sugar. That's all that's going to happen. But instead of happening in 1 step, it's going to happen in a series of like 8 steps. So this diagram is showing you the first few steps of glycolysis over here. Lucky for you, you don't have to know any of these steps. But you will notice, what are these things in green? You can read them.
Enzymes.
They are enzymes. They all end in ACE, hexokinase, glucokinase, phosphohexosisomerase, phosphofructokinase 1. They all end in ACE. So this would be an example of a metabolic pathway, what we talked about in the last chapter. And enzymes are helping catalyze these steps. Now, it says here there's an investment at the beginning. This is the investment right here. ATP is going in and breaking down. This is called the priming of glucose. Basically, it takes 2 ATP to get the reaction started. The rest of this, you're only going to see 1 column on the next page, but you can actually imagine that it's twice, because after this split, there's 2. So you'll see times 2 there. This is the other half of it. Again, a bunch more enzymes. And at the very, very end, this is pyruvate. Pyruvate is just the name of the 3-carbon sugar that's made. So in summary, it costs 2 ATP to start this. That's called the priming of glucose, the investment part. But you make 4. So a question you could be asked on the test will be, how many net ATP are made by glycolysis? 2. We make 2 NADHs. Remember, NAD is a coenzyme. And all that it's doing here is it's picking up hydrogen, which is all these coenzymes involved in cell respiration do. They pick up hydrogens. The hydrogens are going to be really, really, really important later. Right now, it just seems kind of okay, so it picked up hydrogen. That hydrogen doesn't do anything right now. And glucose was split into 2 pyruvates. Again, pyruvate is just a fancy name for the 3-carbon sugar. So we started with this, a 6-carbon sugar, and literally all we did was split it in half. But we did it in a series of steps so that it can occur at normal temperatures and extract some of the ATP. So we've extracted some energy from the sugar. Most of the energy is still here. As a matter of fact, we've only extracted about 2% of the energy from the sugar. Pyruvate still is a large energy molecule. So trapped in these bonds, there's more energy. All right, and the way that we made these ATPs were by substrate level phosphorylation. We discussed that yesterday. Remember, substrate level means that literally we took the phosphate off of a substrate using an enzyme, and we built those ATP one at a time. And I believe there's one more thing here. So it only gets about 2% of the energy from the glucose. A remainder of the energy is still trapped there, which means that if you only run glycolysis, and nothing else, you can make ATP, but only very small amounts. So here's the issue. We can continue to run glycolysis. Let's just say we stopped right there, and we just got 2 ATP from every sugar. We could just keep going, getting another sugar, get 2 ATP, get 2 ATP. There's one problem, is that there's not going to be enough ATP extracted from the sugar. We're not getting enough to support the amount of energy that our cells need. But there's another problem. Oh, this is a diagram just showing, here's the ATP coming in. It splits. You won't have to know any of the names of these. They're just showing you the phosphate, and then we're making ATP at the end, and we get the pyruvates. So here's the problem. If we need NAD to pick up hydrogens in this process, eventually, if you guys were all NADs, and I was sending in glucoses, you guys could be taking the hydrogens and we could be making the ATP, but eventually all of you, NADs, would be holding a hydrogen. Well, that means that we don't have any more NADs to keep glycolysis going. They would all be holding hydrogens. They'd all be, what do we call it when it gains a hydrogen? Reduction. They would all be reduced. We need to get them back to oxidized. We need to get rid of that hydrogen. So if there's no oxygen present, we don't just stop there. Even though we don't have oxygen and we can't go to the mitochondria and extract more energy from sugar, we can dump off this hydrogen so that we can at least keep doing glycolysis. And that's called fermentation. And yes, we do fermentation, but it's not our main way of making energy or collecting energy from sugar. But you're sprinting those last few meters in the race, or swimming the last few meters, when you're swimming, or you're lifting weights and you're trying to hold them up. Anytime you're doing something where you're out of breath and you're not quite getting enough oxygen to your muscles, you don't just collapse. You can still keep going for a little while longer, and that's because you're doing fermentation. So what you're doing, you don't make any more ATP, but you're recycling the NADH back to NAD again. And when you do that, since there's no oxygen, which would normally pick up the hydrogens at the end, some other thing picks up the hydrogens at the end. Possible things that can pick up those hydrogens: ethanol, which is alcohol; acetic acid, which is vinegar, but you won't be tested on that one, don't even worry about it; lactic acid, and this is what we make. The enzyme in our body that we have in order to recycle the NADH is, I believe it's called lactate hydrogenase. It basically recycles the hydrogen and makes lactic acid as the product. And lactic acid is what builds up in your muscles. Anytime you're feeling the burning in your muscles, that is the lactic acid building up because you're not getting enough oxygen to them. Which is okay. It's part of our natural process to keep us going in an oxygen deprivation situation. But obviously only for a short period of time. But it gives you those quick bursts of energy to keep you going. Bacteria and yeast are the ones that make ethanol. If we made ethanol, everybody would exercise. You just get more drunk the more you exercise, so probably make exercise a lot more popular. But bacteria and yeast make alcohol as their byproduct of fermentation. So what we're really doing when we make beer and wine, for example, we're taking bacteria or yeast, we're feeding them sugar. So we're feeding them grapes or whatever the source of the sugar is, and they're just living their lives. They're making ATP because they typically don't use oxygen to do it, so they're going through fermentation. And their waste products, they're sitting in their waste product. Ethanol, alcohol is their waste. And eventually the alcohol builds up enough that it kills them, so they die in their own waste. So see, it's a very cruel thing to make alcoholic beverages. It's also the way we make fresh bread, because there's another product of this also, which you should add to your notes. You do not have to know this right here, or this. This is just showing you what it actually looks like, but I do want you to add to the notes, if it doesn't say it, that the other product that bacteria make, besides alcohol, is carbon dioxide. This is how we make fresh bread. When you make fresh bread, you don't immediately put it in the oven. If you put it in the oven, it won't rise. Because the reason it's rising is the same thing about feeding them and they're making alcohol. You're feeding them the sugar from the dough, and they're in there and they're making carbon dioxide and alcohol as their waste products. So yes, they're actually making alcohol in the bread too. It's a very small amount, number one, and number two, it cooks off when you bake it, so you don't actually taste the alcohol that they make. But the bottom line is that's what's going to make your bread rise. So the longer you leave it there, the more fermentation they do, the more bubbles you get, or the bigger bubbles or whatever. And then when you bake it, basically you kill them. So again, the sad bacteria give up their lives, or yeast, so that you can have fresh bread. So this is what we do instead. We don't make carbon dioxide, we just make lactic acid, also called lactate. So this is fermentation. This is what's going to happen if there's no oxygen present. This is what's going to happen naturally in some organisms, like the bacteria I was talking about. A lot of them, they don't use oxygen at all. They just do fermentation. But it is definitely not the way that we're going to want to go about making our energy. So the other option is, if oxygen is present, which is going to be the normal conditions for us and for plants, other animals, even a lot of single-celled organisms, if they have mitochondria, then they're going to utilize oxygen, and the oxygen will eventually pick up the hydrogens. So NADH, those hydrogens that need to get dumped off somewhere, oxygen will pick them up, and water will be the final product. And when this happens, you're going to get a lot more of the energy from glucose. Rather than only getting about 2% of the energy from glucose, you're actually going to capture about 40% of the energy from glucose, which you might say is still kind of crappy. But if you think of most man-made products, for example, cars, they're only getting about 25% of the energy from gasoline. The rest of it's wasted as heat. So this natural process that may not seem extremely efficient is actually much more efficient than most man-made processes that we've created. And in our case, same thing. The rest of it ends up being lost as heat, which warm-blooded animals kind of use to their benefit as well, really, because we actually use heat to maintain our body temperature, which then actually allows us to have a lot more flexibility and movement in different environments. So warm-blooded organisms have actually kind of evolved a way to utilize this excess heat. All right, the other 3 steps, and we're only going to talk about 2 of them today, are number 1, the oxidation of pyruvate, which has nothing to do with oxygen, it has to do with oxidation. 2, the citric acid cycle. And then the last one, which we'll talk about tomorrow, is the electron transport chain. So those are our 3 steps.